Good afternoon, Warrington. A pleasure to be back to talk to you today. So I'm going to be talking about putting people under the microscope. It's going to be a bit of an amble through some of the work I've done in my research at the University of Liverpool. A lot of it is based on imaging, some of it is based on spectroscopy. And that's the sort of theme that runs through. I'll be jumping around between different topics. I'll be having a start in the world of atoms, because in a sense that's where I started from when I was a postdoc in the University of Birmingham and then came to the University of Liverpool. I was working in a world of atoms, so that's where we're going to start. Then I'm going to take a little bit of an aside and tell you why it is that when we're looking at images, we have to be careful, because what we see in images is not necessarily what's actually there in images. So we're going to be talking about the difference between perception and reality. And some of the work I was doing in those early days has spun off into different areas. Although I work in the physics department, I've applied what I've learned to different areas. So for instance, we'll be looking at earth sciences and then medical sciences. Then we'll actually enter the world of living matter and look at a few different projects that I've been involved with. And the end of the talk will be looking explicitly at putting people under the microscope and investigating cancer through imaging and through spectroscopy. Thanks very much. Okay. So a lot of this talk, not all of it, but a lot of the talk is about imaging and images and looking at images in the scientific context. And so there's two things we have to get established very early to make sure we don't talk at cross purposes. And that is the concept of image processing and image analysis. In a sense, that's what I've spent 30 years of my life doing as a researcher in various universities, is looking at images. Image processing is what you do to an image, what processes you apply to an image, usually to change that image into another image which is easier to see, easier to extract information from. Simply taking an image and changing the colors of the image is, in a sense, image processing. You're doing something to the image, usually for the purpose of trying to see more of what's actually there. Image analysis is a little different. The output of image analysis is not another image which might look different. Image analysis means extracting some numbers from the image. In other words, I want to count how many objects there are there, or I want to determine the size of a particular object in the image. That would be taking an image and producing numbers which come from that image. So that's the difference between image processing and image analysis. One is how to interpret images, the other is how to quantify images. And during this talk, I'll be using a number of different images, some of which I've obtained myself, but many of which have been obtained by people with whom I've been collaborating. And I'll give you a list of all the collaborators at the end, rather than clutter up each of the slides with its origin. Some of them have been coming from colleagues, some from project students. This, I think, is a good place to start because it reminds you just how good microscopes are these days. This is a microscope image. It's a particular type of microscope called a scanning tunneling microscope. And the scale here is, across the image, 850 picometers. And a picometer is 1/1000th of a nanometer, and 1 nanometer is 1/1000th of a micron, which is 1/1000th of a millimeter. So we're talking about 850 times 10 to the minus 12 meters. It is less than 1 nanometer from end to end, this image. And it's showing us a few atoms. In this particular case, these are, I think, carbon atoms, if I remember rightly. So microscopes now can get down to magnification such that we can see individual atoms. And whenever I see a picture like this, I remind myself that if we were to take this particular magnification and ask ourselves how large would a grain of sand be, for instance, at this magnification, a grain of sand would be about the size of the moon when displayed like this. Hence, I always tend to think of William Blake, To See a World in a Grain of Sand. So it's a reminder that microscopes are amazingly good. We've had this technology since about the 1980s, and it's been refined ever since then to get better and better images at atomic scales. So that's a handsome young man in front of a scanning tunneling microscope. The scanning tunneling microscope is actually buried deep inside this vacuum chamber. If we're going to look at individual atoms or molecules, we can't have the atmosphere getting in the way. We can't have the sample being contaminated. We can't have, if we're interested in a piece of iron, we can't have it rust as we try and image it. So we need to basically keep it as clean as possible. And the easiest way of keeping things clean is to put them inside vacuum chambers and pump the air out. In this particular case, the pressure inside is about 10 to the minus 13 atmospheres. So there's a pressure differential on each of these flanges here, 1 atmosphere on the outside and 13 orders of magnitude less on the inside. And then you just hope that this vacuum system doesn't leak whilst you're doing your experiment. These experiments were done inside the Surface Science Research Centre, which is part of the University of Liverpool. And the principle of a scanning tunneling microscope is that if you've got a surface which is nominally flat but is actually made of atoms, and at the atomic level that means we have small bumps in the surface, even if it's atomically flat, that means as you go over each atom you have a little bit of a bump. What you do is bring a sharp tip, as sharp as possible, down towards the surface, and you hold it so that there's a small gap between the tip and the sample. The tip here is represented by these red atoms. Imagine a huge pyramid, much larger than I've shown here. And again, imagine the sample as a huge slab of atoms, the top of which is shown here. If there's a small gap between the tip and the sample and you apply a voltage, no current should flow because there's no contact between the two. But quantum mechanics tells us that there will be a small current that flows, and the current that flows will be very, very sensitive to just how big this distance d is. You could change d by a fraction of an atom, and the current that changes is easily measurable on an ammeter. That's how sensitive it is. It comes from the rules of quantum mechanics that even if you change that distance by a very small amount, you can measure the current, and hence you can effectively measure for a given height of the tip, what does the topography of the sample look like? And using this particular technique, you can get images like this, which was taken by a colleague at the University of Manchester. Notice the scale, maybe you can't read it, that scale bar there is 2 nanometers. And so we're looking at a slab of material. It happens to be the surface of iron oxide. My colleague was interested in oxides, and he was interested to know what does the surface of an iron oxide look like. And lo and behold, it is not uniformly spread with atoms. You don't find iron and oxygen atoms uniformly spread over the surface. Because of the way iron and oxygen atoms interact with each other, they actually form this rather intriguing structure. Each of these little mountains there is 1 atom. It's thought to be an atom of oxygen, and they are bunched together, not randomly, but they are bunched together in this sort of hexagonal array. And notice these islands form at particular spots. The islands themselves are in a sort of hexagonal grid as well. Why would atoms arrange themselves like that? Well, that's what research is all about. Let's look at this surface, let's look at these interactions, let's try and understand why these atoms interact the way they do. What forces are actually making the atoms want to sit in these particular locations? That is a lot of what so-called surface science was about. Given this surface, we might know where the atoms are in the bulk of the material, but what do the atoms do on the surface of a material? How do they interact with other atoms? And this, again, is a reminder that fairly routinely, scanning tunneling microscopes, as they are called, can regularly image individual atoms and individual molecules. Working with scanning tunneling microscope, hereafter simply called STM images, led to the development of some software. My students needed to be able to analyze images that were coming out of these scanning tunneling microscopes. But we had other microscopes as well, such as scanning Auger microscopes. I'm not going to explain all of these, but there are others. Perhaps you've heard of scanning electron microscopes, but there's scanning force microscopes and scanning ion microscopes and scanning laser. There are lots of ways you can image a surface whereby you scan something over the surface to obtain an image. And I wanted my students to be able to look at lots of these types of images, and I realized as I wrote software that allowed us to look at these images that were coming off the microscope and process them and analyze them, I realized that lots of different microscopes needed this sort of software. So scanning tunneling microscope is the one I've already mentioned. But I figured that rather than write a piece of software which is designed to work with scanning electron microscopes and scanning force microscopes and scanning tunneling, rather than having a middle word, let's just call it X, scanning something microscope. You give me a letter of the alphabet, I can probably tell you a type of microscope that uses that letter of the alphabet. So I came up with a totally unpronounceable piece of software which I called Image SXM, simply because the X can stand for A for Auger, or E for electrons, or F for force, or all of these other possibilities. And I've been writing that. I started writing this back in the 1990s. I've sort of now given up. So maybe September 2022, maybe that's the last version of this software I'll ever write. It's been used by a lot of people around the world. It's popular mainly because it's free. If I actually charged for it, it probably wouldn't be quite so popular. But I believe there are many thousands of people around the world now using this software to help them process and analyze their images coming out of these sorts of microscopes. Here's a pin in every country that I think that software is now being used. I'm having trouble breaking into Greenland, and Antarctica seems to be rather devoid of anybody using them. But, okay, there's still time perhaps. And I spent a fair bit of my time in my early days using scanning tunneling microscopes and other techniques, I should say, to actually look at how atoms are arranged. I was particularly interested in a whole range of metals called the rare earth metals, the ones down at the bottom of the periodic table, and I was interested in trying to work out how atoms arrange themselves and why they arrange themselves in particular ways when looked at on particular surfaces. If you take a lump of material and you cut particular surfaces, the atoms arrange themselves in different ways depending on which surface you're looking at. And I was trying to understand that. And eventually, with a postdoc, I wrote a book on how to understand the surfaces of these particular materials, the surfaces of rare earth metals. That's a scanning, tunneling image of a surface. It is clearly not dead flat. Here, the atoms want to arrange themselves in sort of terraces. And the terraces are not aligned at random. The terraces seem to adopt 120-degree or 60-degree angles. And that's because of the hexagonal arrangement of atoms on these particular surfaces. So, if you'll avoid the cliche, if you'll excuse the cliche, I sort of wrote the book on the surfaces of rare earths and thought, I've written a book on it, let's move on and think about something else. I started analyzing other images, and I was particularly keen on looking at images from other people and seeing what I could do in terms of analyzing those images and understanding those images. So one of my colleagues produced an image like this, and he wanted to understand what is the arrangement of atoms in these particular images. This is deliberately not a very good image. My colleague has produced far better images than this, but I like this image because it shows you, even if your image isn't great, what can you do with image processing that allows you to see more in the image? Well, one of the techniques we can use is to ask, what is the so-called diffraction pattern? Another way of asking the question is, what are the frequencies that exist within this particular structure? Do atoms always exist on a particular grid, or do they tend to repeat at particular intervals in space, or if you invert that, particular frequencies? And it's possible to do a mathematical transformation to say, what are the frequencies at which these particular atoms seem to arrange themselves in space, and you get a pattern of dots. The important thing is that pattern of dots is not random, it's telling you about how atoms are arranged in that image. It's very difficult to see atoms arranged in that image. Our eyes find it difficult to pick out patterns like that. But mathematically, we can clearly say there are quite definite frequencies at which those atoms are arranged in space. In particular, we can see there's a ring of 10 of them. That's quite unusual. It's not unusual to find atoms arranged in squares, or sometimes atoms arranged in hexagons, in which case the pattern of spots you would get would be 4-fold or 6-fold. But this is 10-fold. That's very unusual for any material, which is why my colleague was studying these. And what we can do with image processing and image analysis is to say, well, if there is something in that image which appears to have 10-fold rotational symmetry, if you take it and rotate it by 1/10 of a revolution, you seem to get similar patterns back again. Let's ask the question, let's only look at the parts of that image that have 10-fold symmetry. What do we see then? we can effectively do the transformation the other way. And now we see a collection of rather more clearly defined spots. And now we can start to say, well, there appear to be atoms here, 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 here. And we can start measuring and seeing what the relationship is between those atoms. One thing we have to be very careful, of course, is to say, well, are we fooling ourselves? Have we just done something mathematical? Have we just processed that image to the point where this bears no resemblance to what was actually there in the first place? Have we just artificially constructed another image? And so we can ask ourselves by very careful examination, for instance, can you see a sort of daisy there within the blue circle? There's a sort of donut and a daisy petal of 10 petals in that flower. And you could ask yourself, well, that's very interesting. Are the atoms actually arranged like that? Is that why we're getting tenfold symmetry? Or are we just forcing tenfold symmetry onto the image? If we look at exactly the same place in the original image, we think, well, is there anything there? But if you look closely, if you look very closely, perhaps the front row can see it, perhaps the back row can't, there is actually a daisy there with 10 petals, but it's much more difficult to see. So in effect, what we've done is take what is actually there and enhanced it to make it more easy to see what is there. We've used mathematical tools to make it easier to see what's actually going on. And this is what I spent a bit of my time, especially in my first few years when I was in Liverpool. I used to take images and start analyzing them to say, can we actually work out which way round the molecules are oriented on this particular surface? Some molecules seem to be oriented this way and other molecules seem to be oriented that way. Is there any particular pattern to this? I tried analyzing them and came to the conclusion, doesn't look like it. It looks like they're randomly arranged on the surface. And in another case, I was interested that these molecules sitting on the surface aren't quite sitting on a grid that you might expect. It's not simply a nice square grid underneath the positions. They're all slightly off position. Is there any particular pattern to this? Can we tell what the interactions are between these molecules by looking at exactly where each molecule is relative to the underlying grid? That seemed to be not necessarily a dead end, but it didn't quite produce the results I was interested in. But that's the nature of research. You look, you try, you see what you get. In some cases, you get gold. In some cases, you get a dead end and you think, move on, try something else. If we're not looking at atoms, if we're looking at something different, for instance, with astrophotography, in some cases, we don't have to worry too much about doing image processing. If we've got a nice dark sky, like from the middle of Africa, we can take a picture of the Milky Way, comes out beautifully. If we try the same thing from England, we might find we get a whole load of light pollution. And that's the image we get of a particular chunk of sky. And then we might say to ourselves, well, I think the information is in that image somewhere, but I'm having difficulty pulling it out. Maybe I have to do a bit of image processing. So in this case, I used my trusty ImageSXM software package and then started to bring out details that are in that image, but are just very difficult to see. And then by tweaking the colors, you get a slightly better impression. Perhaps you can now see why this object is called the North America Nebula, because its outline bears a striking resemblance to the North American continent. So that's just another example of, once you've got the tools to do image processing, you can apply them to images of atoms, images of molecules, or images of stars. If you're processing images, you're really just processing data. And it doesn't really matter at some level what those noughts and ones actually mean in terms of the image, whether they are representing stars and galaxies, or whether they're representing blood cells, or whether they're representing atoms. And that's why the software I wrote ended up getting used in lots of different areas, not just physics. But before we go any further, let me just tell you why this is a problem. Perception and reality are quite different things. How we perceive images, it's a very interesting whole area, and it deserves a 3-hour talk on its own, but we're not going to do that. But what we think we see in an image is not actually what's there. And quite often we don't worry about that because in day-to-day life it doesn't always matter. But in a scientific context, it can matter. So we can ask ourselves, if we look at an image and we see something, and if we ask the computer to look at the image and it sees something different, which one is right and which one is wrong? In other words, which one is the better image processor? Is it a human brain, or is it a computer? Well, you could argue that they're both computers. It's just that one is made of carbon atoms and the other one is made of silicon atoms. But is there fundamentally any difference between the way we see an image and the way a computer sees an image? That distinction is getting more and more blurred with the advent of artificial intelligence acting in a similar way to a human brain. But let me just give you an indication of how difficult it can be. I hope the color comes across okay. Looks pretty good to me. When we look at these images, we don't see what we think we see. So to start with, is anybody in the audience colorblind? Sorry, it's not going to work for you. It might work for you. It might be that it doesn't. There's a cube on the left, a sort of Rubik's cube, as it were, and a cube on the right. I'm interested in these tiles on the top face of the cube. Ignore the rest. So these tiles on the top face of the cube. On the left-hand side here, how many of them are blue? Does anybody disagree with that number? We've got a few people shouting out 4. Okay. On the right-hand cube, how many of them are yellow? Forget the ones lying in the background, just on the top face, how many of them are yellow? Okay. Are you prepared to put money on that? The 4 on the left, which I presume are these 4, is what you're looking at. And on the right, the 7 yellows are these. Would you be interested to know that the 4 tiles I've just pointed to on the left and the 7 on the right that you think are blue and yellow are actually the same color? Yep, that's right. They are actually the same color. Let me remove all of the other pixels. I'm going to leave those 4 tiles and those 7 tiles alone, and I'm going to remove all of the other pixels in the image, one step at a time. That is what you were actually looking at. You were looking at gray pixels on the left and gray pixels on the right. Your brain basically was telling you you're looking at blue and yellow simply because of the surrounding pixels, fooling you into thinking that they're actually a different color. In a sense, what your brain was telling you was these ones are a bit bluer than every other pixel I can see in the area, and these ones over here are a bit yellower than everything else I can see in the area. Notice the backwash, of course, is yellow in this case and blue in that case. So you cannot believe anything you see. When you see something and you think it's a particular color, it is not necessarily that color, regardless of whether you're colorblind or not. And I was talking to a postdoc some years ago who had spent months looking down a microscope trying to work out whether the center of this cell was blue or gray. And he would be counting them. That one's blue, that one's blue, that one's gray, that one's gray, that one's blue. I showed him this, and he was totally deflated because he realized that he had spent 3 months that were completely subjective and didn't have any scientific basis. You look at this and you say, okay, color can be confusing, but surely humans are good at telling this is brighter than that. I mean, that must be easy. Color, maybe that's a bit subtle, but brightness, that should be easy. So here we have a square that's labeled A. I'm not interested in the letter, I'm interested in the square on which the letter sits. There's a square labeled A and a square labeled B. Which is the brighter of the two? I know you're getting very cautious now. You look at that, and you're fairly convinced that square B is obviously brighter than square A. Is it twice as bright? Not sure if it's twice as bright, maybe. But then we can do the similar trick. I'm not going to remove any pixels. I'm just going to add a blank mask to the left, and I'm going to add a blank mask to the right. Now look at the shade of gray. It is exactly the same shade of gray. It's the same shade of gray as the ones on the left and the right. Not similar, it's exactly the same. I checked it with a computer and made sure those pixel values are identical. So they are quite definitely the same shade of gray. So now you know that. So let me take the masks away again. Right, so you know that A and B are the same shade of gray. What is your brain telling you? Even when you know the answer, it is almost impossible to overrule the fact that your eye and your brain, working in tandem, are giving you messages that you can virtually not override. You just have to accept that your brain is trying to do the right thing. Your brain is trying to make life easy for you by telling you what it thinks it you need to know. But unfortunately, that is not scientifically the case. It is not accurately the case. It is not quantifiable in terms of the actual pixel values. So that's why we have to be very careful. We sometimes look at images and we say to ourselves, well, this should be an easy thing to analyze. Somebody said, I want to analyze how many little squiggles of DNA there are. These are strands of DNA sitting on a glass slide. Glass is smooth at the level that we look at it, but if you look at it with a scanning tunneling microscope, you realize that it's actually a little bit blotchy, it's rough, on a scale of microns. But you look at this and you say, well, I can clearly see the DNA, that's obvious. And therefore, if I asked you, I'm not going to, but if I asked you how many little squiggles of DNA are there, you would simply say 1, 2, 3, 4, 5, 6, and you would say there are 25 squiggles on that image. Easy. 5-year-old could do that, no problem. But if you ask a computer to do it, then it's altogether different. If you ask yourself, well, if I were to take an intensity or a brightness variation as I move across the image, what would I expect that intensity variation to look like? Well, it's obviously going to be a little bit of noise in the background, and then I'm going to go through a bright strand of DNA, and then there's going to be a little bit of noise, and then another bright DNA, and then a little bit of noise, and another. So that's sort of in your head what you think brightness as a function of position probably looks like. Well, actually, it looks like that. And I challenge you to figure out which particular line have I drawn to give me that particular variation, and which of those bumps there correspond to DNA, and which of them correspond to just random fluctuations in the background. It is impossible to tell from an image like that. The actual line I took was that red line. And so that is the intensity variation, and then I get to a strand of DNA, and then another strand, and then a third one. So there are 3 DNAs in that particular profile. Which particular peaks do they correspond to? Well, they correspond to that one, and that one. No way you would have pulled that out from looking at the brightness. So they are not the 3 brightest bits on that particular profile. Your eye and your brain are doing far more image processing than we normally give them credit for. They are taking an enormous amount of information and deducing that these squiggles, even though they are not the brightest things in the image, these squiggles are the squiggles we're interested in, and everything else is noise in the background. And that's the result of hundreds of thousands of years of evolution of your eye and your brain figuring out what it is that you need to get out of these images. But in this case, when I was approached by somebody who said, I want the computer to do this automatically, well, it is a challenge. How on earth do you pull the information out when the profile looks like that? I had to find a different way of approaching the problem. I actually said, well, I'm going to process that image in such a way, no details here, such that the DNA becomes even more obvious. It actually is going to be black with a white background, and now I can easily extract the information I want. But of course, it takes a certain amount of effort and quite a few cups of tea before I could figure out how to process that image in such a way that I produce that image, and as soon as I've got that black and white image, now it's trivial to actually extract there are this many strands of DNA and their average length is this much or whatever. So let me briefly just talk about a couple of spin-offs before I get into the world of living matter. Along the way, I've realized that there are applications. If you look at analysis of images, there is no reason why you have to restrict it to anything that I did in my early research. If you've got an algorithm that's capable of separating the wheat from the chaff, doesn't matter whether it's separating DNA strands from glass background, or whether it's separating blood cells from the serum, or whether it's separating these stars from these galaxies. It doesn't matter what the original algorithm was, it can probably be applied to other areas as well. And so I started realizing that some of the work I'd done can be applied in earth sciences, and more recently I've realized that there's a lot of areas in medical science that this can be applied to. So it took me a while, but I came up with these acronyms. The acronym is the most important thing of any research project, of course. Computer Integrated Polarization Image Analysis, Principia, and Microscopy Image Analysis Software for Medical Applications, or miasma. The medics loved that one. So let me just briefly say some of the work I did in collaboration with a real earth scientist I was only playing at it, but I collaborated with a professor in Switzerland who basically was an expert in looking at rocks. And between us, we came up with algorithms by which we could analyze what rocks look like, what different minerals look like, especially in a microscope that's got polarizers which color the rocks in particular ways according to the orientation of the atoms within the minerals that we were looking at. We came up with a way of doing the analysis in order to give us quantitative ways of looking at how the particular minerals were structured. And between us, that resulted in my second book on image analysis, available from all good booksellers, by the way. But in terms of time, I spent more time looking at medical applications, and this was a particularly rich area for getting project students involved. I had some expertise in image analysis and image processing, but I could then say to project students, right, your job is to look at these images and figure out what the parasite is doing in this particular set of images that's been obtained from the local hospital or something like that. And then they would learn a bit of image processing and image analysis whilst they went through their project of actually trying to extract some information from the images they were given. So that was the line we went down with looking at particulates, meaning how much carbon is there in these lung cells, looking at biopsies taken from people's lungs to see to what extent smoking had damaged their lungs because of the carbon particulates that existed in the cells, looking at leishmania parasites and malaria parasites, looking at I'll come back to that in a little more detail in just a moment. Looking at retina, perhaps you're used to the idea that you go to an optician, they can photograph the back of your eye. One of the reasons they do that is because it's not only telling you how good your vision is, more importantly, it's telling you, have you got any early signs of macular degeneration? Have you got any early signs of diabetes that show up in terms of the structure at the back of the eye? So trying to identify what is a normal retina and can we identify any abnormal areas of a retina was again an interesting area, collaborating with the ophthalmic surgeons within the university. Looking at the morphology of different parasites, parasites change their shape depending on how long they have been in the animal or human that they are infesting. If you look at a particular tissue sample and look at the parasites, from the morphology of the parasites, it's possible to tell, are they a month old or are they 3 weeks old or 2 weeks or 1 week? At what point did this animal get infected? So morphology gives you life cycle in a sense. I was looking at lymphocyte cells, looking at lipid bodies, looking at microfibrils. These are all structures that may or may not be interesting to the person who generated them, and looking at structures of bacteria to see how bacteria grows. One of the more interesting ones that I did, which turned out a little different than I thought, was looking at human chromosomes. The Liverpool Women's Hospital regularly do chromosome screening of everybody that comes through the system, as well as acting as a screening centre for women all over the UK. So anybody that gets pregnant ends up with chromosome samples taken at some point during their pregnancy, and then an expert looks at the entire chromosome set, the so-called karyotype, and tries to figure out, is that normal? You've got to have respect for anybody that can look at an image like that and within minutes can say, everything's fine. Or within minutes can say, hang on, that doesn't look right. And they can actually, using the banding of these chromosomes, there should be, of course, 23 pairs of chromosomes. Each of the chromosomes has a particular banding pattern. This is the sort of cartoon of what the chromosomes of a human should look like. There should be particular banding patterns for each of the 23 pairs, including the XY pair that determines the gender. But the question is, can this be automated, or is the only option to take photographs, put them in front of an expert who then spends hours poring over these images, trying to work out whether these chromosomes are perfect or whether there's any chromosome abnormality, which would either tell you there's going to be a problem with the baby, or is it worth terminating the pregnancy early? Being rather naive, I thought, yeah, that should be doable. I don't see why we can't write some code to do that. But the more I tried, the more I realized this is a really intractable problem. not least because it is very difficult to get all the chromosomes completely separated. So notice that a lot of them overlap with each other, which means the banding pattern on one can be seen, but the banding pattern on the other is sitting underneath. And so you can't actually tell what's going on when two cross. And there's nearly always a crossing point. So you take multiple pictures from multiple cells, in which case chromosome 13 will be over here in this case, but it will be somewhere else in the image in the next image that you take from the next cell. And how can you tell that's chromosome 13? How are you going to actually identify each of the chromosomes? In principle, they should be different by length, but look how close, for instance, chromosome 9 and chromosome 10 are. They differ by the tiniest amount in terms of length, but an expert can tell which is which by looking at the banding pattern. So it's a roundabout problem. If you had the banding pattern, you could work out which is which. But you can't work out which is which until you've got the banding pattern, and you can't get the banding pattern until you've figured out that this chromosome is the same as that chromosome, because in this image it's blocked underneath another one, whereas in that image it isn't. So you've got to look at multiple images and identify which chromosome is which across multiple images to get the entire so-called carrier type, the entire set, and only then can you say, yep, I'm quite convinced that all the chromosomes are perfect except for chromosome 9, that's got a problem. And then what disease is going to result due to an aberration on chromosome 9? That's determined by geneticists. And it turned out to be much more of a problem than I anticipated. I suggested it was a project student that tried it. They tried it and they got a certain progress. They managed to go a certain distance with this. But we never got to the point where we could definitively say, give me a set of images, I can tell you definitively whether this chromosome set is perfect or whether the chromosome set is imperfect, and there are chromosome abnormalities. So as far as I know, they are still doing this manually with a bunch of experts who are trained to recognize what is a perfect chromosome set for a human, and they can basically spend I don't know how many hours a day just staring at these images and coming to the conclusion that one is okay, that one is a problem, that one is okay, that one is a problem. So it's an interesting problem, and I thought naively that maybe automating it would be simple enough that within a year or two we could have a piece of software that would do it automatically. Never came to pass. A very interesting project, a very good way of training up students, but it didn't result in a piece of software that we could then sell to the NHS and say, here's your problem solved. You can now go and sack all the people that were looking at these images. Of course, you need experts to make sure it's right in the first place, but ultimately, could we save them time was the question. So let's move into the world of living matter. I'm going to take two particular areas that I've been involved with in past years: microcirculation analysis and cancer. And I want to give you just a taste of some of the issues trying to deal with these two problems. So I've got, 10 minutes each on something like that. So microcirculation, this was a consultant at Alderhay Hospital who was working with kids, trying to work out when sepsis was going to be a problem. In other words, when you have blood poisoning, if you want to give it its old name, when you have sepsis and meningitis causes sepsis, when you have a situation where the blood circulation is affected, the actual degree to which blood is not circulating in the microcirculation systems in the body, that can be an indicator that you have sepsis, which might be an indicator of meningitis. At the very least, using circulation within the very small blood vessels, the so-called microcirculation, that will tell you whether or not the treatment you are giving to meningitis is working. So you might have a patient for which you know what the disease is. The question is, if I give them this drug, do they get better or worse? And I want to know within a few hours. It's no good giving them the drug and then finding a week later, no, that wasn't the right drug to use for this particular situation. You need to know very quickly whether or not that drug is working and improving the microcirculation, or whether the microcirculation is getting worse, or at least know better. So you want a fast way of being able to analyze the circulation to know how to treat meningitis patients. And that was done with a small portable microscope that the clinicians use. And it's a small microscope. It's not much bigger than an electric toothbrush. And it goes under the tongue. So what we're looking at is that, the underneath of the tongue, or the tissue that's connecting the tongue to the base of your mouth, so-called sublingual area. So it's only imaging a small area under the tongue because that particular part of the tongue allows you access to the microcirculation. It's difficult to see blood vessels when there's a load of flab and fat and other things around, whereas that particular point, easily accessible under the tongue, gives you very close access immediately to the microcirculation bed that exists in your mouth. So it's not so much putting people under the microscope, it's more putting the microscope under people. And this is what we're dealing with. We're dealing with microscope images, strictly a video, which might be 20 seconds long or something. So a consultant would go to a patient, they'd want to know what the microcirculation is, so they would put this microscope under the tongue, they would image something like a millimeter or so, a millimeter by a millimeter, so it's a very small area being looked at, and they would obtain an image like this. This is a particularly good one in the sense that it's quite steady. You can see what's going on. You can see it's effectively a bed of capillaries. Of course, these are very small because that's a millimeter, so some of these are barely micron in size. But one thing you might not appreciate unless you look at this very carefully is you cannot actually see any blood vessels at all. What you're looking at is blood cells. So you're looking at individual cells that are following a particular path. They are following the paths of the blood vessels, but the blood vessels themselves do not show up because this particular microscope is looking for hemoglobin, so it's looking for the blood cells. So you might think that you see capillaries, but actually you're just seeing effectively the ghost of individual cells moving. And perhaps you can see that some of them don't move very fast. Over here, they seem to be quite sluggish. Up here, they seem to be moving quite fast. Down here, they seem to be moving quite fast. There are some here that seem to be stopped completely. Perhaps you can see individual cells here that don't appear to be moving in one direction or the other, regardless of the heartbeat of the patient. So that's what microcirculation analysis means. The consultant said to me, at the moment, we generate these images, these videos, and then we have to have somebody sit down and watch them. And it takes hours and hours and hours for somebody to sit down and look at lots of these videos and make the judgment call, this is good, this is bad, this is not so good. Is it possible to quantify this, they said? Is it possible to quantify the motion, the circulation, how fast are these blood cells actually going through the capillary net? And is it the same over the entire network? So I said, okay, that should be doable. But I hadn't appreciated that that is a very steady video, and most of them are much worse than that, because if you think about it, you're putting a microscope underneath somebody's tongue and you're asking them to stay absolutely still for 20 seconds. If you look at what's going on within one particular part of it, you might say, well, I can't really see what's going on. Let's boost the contrast a little bit. Well, there does seem to be something there. But then if you say, well, what's actually going on? Are any of those blood cells? How can you tell? Some of them appear to be a bit darker than the others. But we're trying to work out the motion of something we can barely see relative to a vessel that we cannot see. The vessel itself isn't imaged. So they're asking us, can we quantify the motion of a blood cell relative to a vessel? And that vessel, in principle, will appear to move. Of course, if the microscope moves, then the whole capillary network will appear to move. Any attempt at quantifying will have to deal with lots of different things. It'll have to deal with translation of the microscope, or perhaps the tongue relative to the microscope. The magnification might change, because that depends exactly where you have the microscope relative to the patient's tongue. How can you be absolutely sure that you're not rotating the microscope, or that the tongue is not rotating, or there's any distortion? The tongue is not a rigid object, it's flexible. So the image you're looking at could, in principle, this network of invisible capillaries could, in principle, drift across the image. Or the magnification might change, in which case the actual size of the capillary network changes. Or it might actually rotate during the video, in which case the entire thing will rotate round. Or there could be distortion of different tissues stretching or collapsing. Or more likely, there's going to be all of the above. So how are you going to look at an invisible network and tell what the network is doing so that you can then say what individual cells are doing relative to that network. And that took me ages to figure out. I had to find different ways of effectively stabilizing the video. In other words, the video will jump around and it will change and it will distort. Is there a way of processing those images to try and get them all to line up? If we can do that, then we can start to say that cell appears to be not moving relative to the network, or these cells appear to be moving quite rapidly. And of course, it's not just those, it's also the fact that the patient might be on a ventilator, for instance, depending on how ill they are. If they've got meningitis, they may well be on a ventilator. And of course, there will be a heartbeat changing the way the cells move. So I used a number of different techniques, I'm not going to go into the details, to try and work out what the cells were doing down in these microcapillaries, anything larger than about 7 microns. 7 microns is about the size of a blood cell, so that's the smallest capillary that can allow blood cells to flow. And I ended up quantifying a number of different videos that were given to me from these patients appear to be healthy, and they also gave me a whole load of videos of these patients are quite sick. Can you tell the difference between the two by measuring the blood flow? And I found that the blood flow through healthy individuals was something like 500 microns a second. The blood will flow at the rate of about 0.5 millimeter per second in these capillary networks, and it will go backwards and forwards as the heart beats. Whereas in sepsis, if you have a patient with sepsis, the average velocity of blood flow will be somewhat lower. Also notice there are no capillaries with 0 blood flow. In a healthy individual, all capillaries will be perfused. They'll all have some blood flow. Whereas if you have sepsis, some of the capillary network we look at will have no flow at all. The flow will have stalled. So I went back to the clinicians, the consultants, and said, you asked me to quantify the blood flow in humans for this sublingual microscope. I seem to think that according to my calculations, it looks like the average flow of blood in a human capillary network is approximately 500 microns per second. Can you tell me if I've got the calibration right or wrong? What is the correct answer? At which point the consultant said, I don't know. Give me a video, I can tell you if it's a thumbs up or a thumbs down. But no, I don't know of anybody that's actually measured what this flow actually is, because it's a difficult problem. And to them, it was a don't care as long as they can tell the difference between a healthy individual and an individual with sepsis. That was a bit of an eye-opener for me, that as a physicist applying a computer code to try and get an answer, I realized that there is no answer that is correct, right or wrong. It's useful to the consultants to know the difference, but they couldn't tell me you've got the calibration right or the calibration wrong, that it should not be 500, it should be 300. They didn't know those numbers at that sort of precision, but they did know what to look for. So in principle, this system could be used to automate the process, to take some of the time off the consultant's hands. It's not going to replace the consultant, because if you've got a problem like this on the right-hand side, then of course the consultant would then review that patient and double check, yes, you're right, this one is in serious trouble, we need to get them off this medication and onto a new regimen of medication. So that's work I was doing with aldehyde. In terms of an output, it's not a yes or no output. I thought the easiest thing to do would be to actually color code the network according to how well perfused it is. If blood is not traveling very well through the capillaries, it would end up colored red with a very low perfusion value, very low velocity through the capillary. Whereas anything that's reasonably well flowing at 500, 600, 700 microns per second would be colored light green or perhaps blue. So that would be a healthy individual. But if you colored the same map for a person with sepsis, you would get a lot of deep reds and oranges, indicated that this person's capillary microcirculation is very poor, and you need to look very closely at what's wrong with this patient. So I figured that was the best way of giving an output, not to give a whole load of numbers, not to give a bar chart, not to give a pie chart, but to give a colored indication so that in principle, anybody can use that microscope with a little bit of training to keep it as steady as possible. And then the software would give you a color map telling you roughly what, if any, is the problem with that circulation. And then you can go and consult with other people if necessary. So that was something I did in collaboration with the NHS, in particular at Alderhay. Let me finish in the last 10 minutes or so with a look at cancer. This is partly imaging and partly spectroscopy. Because we're dealing with microscopes, we tend to call it spectromicroscopy or microspectroscopy. We're dealing with imaging with microscopes and we're dealing with spectra. And this is the result of some work we've been doing in the last few years, getting funded either from the Engineering and Physical Sciences Research Council. They tend to fund a lot of physics research, but more recently, Cancer Research UK and the National Institute of Health Research have been funding us to do various investigations, various sort of projects involved in, if we look at images, if we look at spectra, are we able to get a handle on cancer diagnosis, especially early diagnosis of cancer, and can we get a handle on prognosis? Can we tell what is going to happen? Not, it's already happened, it's already a tumor. Can we actually say this is likely to become a tumor or this is likely to stay benign for the next N years? So in other words, we're asking, can we use, and we're interested in infrared, can we use the absorption of infrared light as a sort of signature for tissue that is likely to become cancerous? Or if you like, can we answer the question, can we detect cancer before it's cancer? There's lots of ways of detecting tumors. The question is, if we take tissue that looks healthy, can we tell that that is likely to become cancerous? That's a rather trickier problem. Getting prognosis sorted is much more difficult than getting diagnosis sorted. Why do we use infrared? Well, we use infrared because, again, not too many details, but if we look at the frequency of infrared radiation, infrared wavelengths, let's say, around 10 microns or so, a wavelength substantially longer than visible light. If we look at those sorts of wavelengths, the infrared frequencies are very similar, or in many cases the same, as the frequencies with which various molecules vibrate, especially biomolecules. So in other words, there are lots of molecules in the body which vibrate at the same frequency as infrared light. So if we shine infrared light onto a tissue sample, then if the frequency of the infrared matches the vibrational frequency of the molecule, it'll start to vibrate and it'll absorb some of the infrared light. So we can use the absorption of infrared as a way of saying, well, if that particular wavelength has been absorbed in this tissue, then that particular molecule must be present. It's not quite that simple because lots of molecules absorb infrared at the same wavelength. But in principle, it's if infrared gets absorbed at this wavelength, that means this molecule appears to be present in the sample. So we can use the infrared absorption as a way of identifying what's inside the sample. So we started effectively just down the road, Daresbury Laboratory. We started experiments using an infrared light source that was part of a particle accelerator. It was called ALICE, and it was a particle accelerator in which electrons were put into this system, and basically, through a mechanism which I'm not going to describe, it produced infrared light, and that was a very intense source of infrared light that could be tuned across various wavelengths. If you simply get a laser like this one, this laser only works at one wavelength. It's red, and that's all you're going to get. We wanted basically a intense source of light in which we could change the color over an infrared range that we thought was interesting for particular molecules. So that was a very good way of starting our research, but is a very expensive way of getting infrared radiation. Taking infrared radiation from a free electron laser that's part of an accelerator, it cost millions of pounds, and we were riding on the back of other projects to try and use this light before this particular accelerator was closed down. But more recently, we realized that there are ways of doing this in the laboratory using what is now known as a whole range of different tunable lasers. Unlike these things, which only work at one particular wavelength, you can now buy lasers that can be tuned across a range of infrared frequencies, and that's what we're now using. So that particular way of generating infrared, So it's not obsolete, but this is still a way that you generate radiation of various kinds, whether it be infrared or X-rays in other synchrotrons, other accelerators around the world. If you take a beam of electrons and wiggle it by sending it through lots of north-south magnets, so it gets diverted to the left and then the right and then the left and then the right, if you can make it wiggle, that wiggling electron will produce radiation. Basically, if you take a charged particle and accelerate it, you'll generate light of one wavelength or another. In this particular case, it wiggled at just the right frequency to generate infrared light. So that's how we did it in the first place. We sent the light bouncing from the free electron laser through a number of different mirrors onto a microscope. This is another example of a scanning something microscope. This particular one is called a SNOM, a scanning near-field optical microscope. Again, we're not going to worry about the details. It's just another way of doing microscopy using infrared radiation. And we asked ourselves, if we radiate a piece of tissue at different wavelengths, do we see anything different, or does it look the same at all wavelengths? Very difficult to tell with our first data, because the data was rather poor quality, not very high resolution, lots of noise, difficult to see what on earth was going on. Even with a little bit of processing, it was difficult to see what we were looking at, but it did give us the indication that maybe we're on the right track, because this does not look the same as that, which means this particular chunk of tissue is not absorbing this wavelength in quite the same way as that same tissue is absorbing that wavelength. So there are some differences for a given piece of tissue that depend on the infrared wavelength. So it is a rather tentative start, but it told us it was worth continuing the research down this track, because it does look like infrared absorbs at different wavelengths according to tissue type. And we followed that up by looking at cancer cells to try and identify whether or not we can see particular molecules that are present or not present in cells that are known to be cancerous or cells that are known to be benign. And so we started down this line, again, using microscopy to look at the structure of the cells and using infrared spectroscopy to ask ourselves what molecules are present in those cells. And we end up with a combination of the two, spectromicroscopy or microspectroscopy. We end up taking data which is in X and Y, we're looking at a particular tissue, imaging it in the spatial sense in X and Y, but in the Z sense, this particular cube, as we go back, that's looking at different wavelengths. So you can either think of this as being lots of different images, each image like a deck of cards, each card is an image, each one taken at a different wavelength. So we end up with maybe 1,000 cards in our deck of cards, each one is a different wavelength. Another way of thinking of it is to say that each pixel in the image actually is not a single pixel, it's actually an entire spectrum. And the spectrum is telling us what is the infrared absorption as a function of wavelength. You can't see the scale here, but the horizontal scale in this top right spectrum is wavelength. So it tells us that some wavelengths absorb a lot, some wavelengths absorb a little. You might think that looks fantastic to have lots of variation. The trouble is, healthy tissue looks like that, and cancerous tissue looks like that. In other words, they look very, very similar. If you think about it, cancerous tissue and ordinary healthy tissue both contain lots of the same molecules. They both contain lots of lipids. They both contain lots of proteins. They both contain lots of DNA. And the subtleties of what is it in those tissues that will allow you to say this will become a problem, that won't become a problem, sometimes those differences are absolutely tiny. And you can't just look at the spectrum and say, oh, there's a peak there, this is obviously going to be a problem. Much more subtle than that. We basically have to get machine learning on board. We have to get computers to learn what is the difference between two spectra that might look very similar, but one of them is from a healthy patient and one of them is from a patient who did develop cancer 1 year later. In other words, we use historical biopsies so that we know the history of the patient, and then we can teach a machine learning algorithm to recognize what the differences are. So can we use infrared absorption to tell what's going on? Yes, we can. The work at DESRI showed us that this was a possibility, and the last few years we've shown it definitely works. One of the developments from one of the PhD students working at Liverpool was a little bit of a breakthrough in the sense that we know that infrared wavelengths generally are much longer than visible wavelengths. And this is a problem. Perhaps you've heard it in the context of the James Webb Space Telescope. If you want the resolution of an optical system, you need to know how big is the lens or the mirror and what is the wavelength involved. So for instance, James Webb Space Telescope has a much bigger mirror, which should mean higher resolution, but it's working at much longer wavelengths, which means lower resolution. That's why the resolution of the James Webb turns out to be roughly the same as the Hubble Space Telescope. And it's the same problem with microscopes working in the visible and working in the infrared. We can get much better spatial resolution with optical microscopes because they have short wavelengths, whereas the infrared, much longer wavelengths, we are going to suffer from not being able to see the same detail. You can't really see it in these images, but this is just showing you the process by which we've decided that, in principle, we can fuse the information in an optical image, which is at very high resolution. We can fuse that with the data from an infrared microscope showing us which molecules are present. The information here is rather poor in terms of spatial resolution, but we can fuse information from the two to get an output. I can show you that in a little more detail. An optical image will show us in great detail what cells exist, but it can't tell us what's going on in those cells. We've got no idea what molecules are present. All we know is there's a cell here with a nucleus and there's a cell here with a nucleus. Information with infrared tells us what is actually going on, what molecules are present. And so the different colors here are telling us about different molecules that exist within the material. And the beauty of what this student has done is by combining, fusing together information from the optical at high resolution, fusing it with molecule information at lower resolution, you can combine them together and end up with the image on the right, which has all of the spatial resolution of the optical, but it also has all of the molecule information from the infrared. This, we think, is a fantastic step forward to be able to do this. It's the result of processing techniques that have been developed over past years, but applying it in this way gives us the best of both worlds. The best optical resolution plus the best molecular information, combine the two together and we get wonderful information about what's going on inside materials. What are we doing now? I better wrap up in the next few minutes. What we're doing now is looking at oral cancer, looking at epithelial cells. An epithelial cell, not just your skin, when we're talking about covers all surfaces of the human body. We're not just talking about external surfaces. For instance, the surface of the esophagus or the surface inside the mouth. Inside your cheek are epithelial cells because that is where you meet the rest of the world, as it were. And so we've been studying to some extent esophageal cancer, but now we're looking in particular at oral cancer, explicitly this particular thing, oral epithelial dysplasia, meaning if there's something that looks wrong in your mouth, if a dentist looks in your mouth and sees a little lesion on the side there, they might say, is that something to worry about or not? Is it worth referring you to a specialist, or is that something you can just ignore? Our aim is to develop a device that will use infrared absorption to tell you whether or not that little lesion in your mouth is something that you need to keep a very close eye on because it looks like it could be a problem, or whether we tell you, nope, that's benign, that's not going to do anything. We can perhaps check it every couple of years, but it's not going to do anything. Whereas if it looks worse, perhaps you need that checked, and maybe you need a close eye on that every 6 months just to be on the safe side. So that's what we're currently working towards, improving the accuracy, not necessarily of diagnosis, but the accuracy of prognosis, being able to tell what is going to happen in the future, not tell you it's already too late, you've got mouth cancer, but this is definitely worth keeping an eye on because this could become a problem in later life. So we're trying to set up some way of spinning off this technology. We think we've got an interesting way of analyzing the data. We think we've got an interesting idea to how to build a device that will actually do this for mouth cancer, and who knows, maybe it could be applicable to other cancers as well. So we're trying to set up a spin-out company whereby we can actually start to develop this further. We're thinking of the Liverpool Diagnostic Infrared. but I like the idea of calling it the Elder Wand just because I'm a Harry Potter fan. So it would be nice, and the original origin of this, if you'll excuse the tautology, the origin of this was a surgeon saying to us, wouldn't it be great if I had a handheld device such that when I did surgery, I could just wave it over the patient and I could tell whether or not I've got all of the cancer or not, or whether there's any tissue left that could, in principle, become cancerous in the future. Can I tell that I've got it all or not? So it was a surgeon who said, is it possible to build one of these things? And that's what got us started thinking about what can a physicist do to identify molecules in tissue that could in principle be useful for diagnosis and prognosis of cancer. And that's where the idea of the Elder Wand came from. And ultimately, it would be great if we could find enough funding to build one, to put it into the hands of the surgeon who could then use it as an in vivo tool. At the moment, we're thinking of doing it purely on biopsy samples, on tissue samples, but in years to come, wouldn't it be great if we had a diagnostic and prognostic indicator that a surgeon could use so he or she knew exactly what they were doing? So I've been trying to take you through this 30 years of my life, starting by imaging atoms, imaging molecules, developing it into earth sciences, a little bit of an avenue, and then developing into medical science. No science is ever one-man show. These are the collaborators, mainly at the University of Liverpool and a few elsewhere as well. And of course, we are grateful to those people who funded our research over past years. Thank you all very much.